Great. Thank you, Richie. And it was about that time that I met Bill Kern. Bill, I think I met you probably about 1988 or 89. But I'll just read a little bit of your bio here, Bill. Dr. Bill Kern is an associate professor in the Department of Entomology and Nematology at the University of Florida and is based in at the Fort Lauderdale Research and Education Center. He received his BS in Life Science and Chemistry and his MA in Life Science from Indiana State in Terre Haute, Indiana. He got his PhD in Entomology and Zoology from UF in 1993. Probably met you 3 or 4 years prior to that, Bill. He served as an assistant extension scientist, the Central Florida Wildlife Extension Specialist with the Department of Wildlife Ecology and Conservation at UF from 1993 to 2000. He served as an assistant professor from 2000 to 2007, and then an associate professor from 2007 to present in the entomology department in Fort Lauderdale. His areas of expertise are urban entomology, Africanized honeybee management, vertebrate pest management, nuisance wildlife management, and medical and veterinary entomology. And on a personal note, I think I've known you about 35 years, Bill, and we first met, me and Lisa were taking a vertebrate zoology class, and you were the TA for that class.
Yeah, I remember.
You remember that? And you were looking for a summer job. I said, Phil Kaler out at the USDA is looking for technicians. Nancy Hinkle was out there at the time, I think. Nancy's online. Yep. I just saw Nancy just chimed in. She said hello. And you came out there, and kind of the rest is history. But one of my favorite stories of the vertebrate class was you took us on a field trip. I don't know if you remember this, but folks, if Bill's one of the best naturalists that I know, and we saw a black racer. This was out in Paynes Prairie. Do you remember this? And you ran over there and grabbed the black racer.
Yeah.
And you grabbed it right behind the head, too far behind the head, and it turned around and latched onto you.
Yeah.
And Bill is sitting there talking to the class about the identifying characteristics of a black racer, and its mouth is clamped on your finger. There's blood running down your arm, and you're talking about how to identify a black racer. That's a vision I have 35 years ago. But anyway.
Were you there when we were out again at Paynes Prairie? And I told the class, watch out, because I smell a cottonmouth nearby.
No, I wasn't there.
And so I'm looking around, I'm going upwind, and sure enough, there's a cottonmouth on the bank, and he had just thrown up a Florida round-tailed muskrat. And he was not happy with us. That's why he was musking so bad.
Okay. All right, I'll go ahead and turn it over to you, Bill.
Okay. All right, let's see. All right, can you see my screen right now? Because I'm looking at you, Dan.
Yeah, I don't see your screen. Does he have to share screen again, Richie?
Yeah, Bill, you'll have to,
to
go ahead and click on that share screen again, since I rudely stole it from you earlier.
Okay. All right. Everybody see it fine?
Yep, looking good.
Okay. Good afternoon, everybody. And today I'm going to be talking about those insects that bite and sting. And one of the things that Dan didn't mention is I did my PhD on fleas. So you're probably going to learn more about fleas than you really wanted to know. But let's go ahead and get started. And one of the things the first part on the fleas is actually part of a program that I put together for, I can't remember if it was for the University of Georgia or the Georgia Pest Management Association, or it might have even been for Southeastern Branch. But it was the diversity of urban fleas and their natural history in the Southeastern United States. And it's largely based on a couple of publications. This is Fleas Parasitizing Domestic Dogs in Georgia. And this is the information about it, if you want to look it up. But it had a lot of good information about which flea are being a problem in the Southeastern United States. So this was for Southeastern Georgia. And one of the things you'll notice is, which is always the case, is that cat fleas, Tinocephalides felis, is the most common flea on dogs and cats. And 61% of all of the fleas that were collected were cat fleas. You also get a fair number of dog fleas, but usually those are going to be from rural areas, especially areas where you have a coyote population. Pulex simulans, the southern human flea, is third most common. And then finally, the fourth most common is the stick-tight flea, Echidnophaga gallinaceae. So these are the primary fleas that we're going to be dealing with. And then these others, the remaining two are the cottontail flea, the spiny rabbit flea, the eastern squirrel flea, and then the cotton rat flea. And those are all accidental that get on dogs and occasionally on cats. So also in this study, they were looking at the seasonal distribution of fleas on dogs in Georgia. And so the months that were selected are those months where there were over 100 fleas collected. And of course, we have more months represented because we had a lot more cat fleas than anything else. So basically the summertime from July through November, when we start to get into November, and we start to get cold spells, then we're going to see outdoor flea populations crash. In Florida, we actually see much more cat fleas inside homes almost throughout the year because we have such high relative humidities. The dog flea, we just have it from September and October. Those are the only 2 months where we collected over 100 fleas. And again, partly that has to do with the dog running season. Hunting dogs are going to be out where they're going to be in the same habitat as coyotes. And coyotes are the source for dog fleas in the southeastern United States. And then Pulex simulans, which is the third most numerous, was primarily seen in August. And it probably had a wider distribution, but we had less than 100 fleas collected. Another piece of work that I cited was the prevalence of ectoparasites in the population of feral cats in north central Florida during the summertime. And again, there is the information if you want to look it up. And so they were collecting the fleas from feral cats in north central Florida. So that's largely from, say, Ocala north to the Georgia border. 92% of the fleas collected were cat fleas, which is not too surprising. Only 4%, 4.5% were the southern human flea. And that really isn't a good name for Pulex simulans. Pulex simulans has a very broad host range. It doesn't just feed on people. It feeds on all kinds of large and medium-sized animals. And then the third most common was Echidnophaga gallinaceae, the stick-tight flea. And oftentimes when you find stick-tight fleas on dogs and cats, they're very often around the ears, and you'll often find lots of them. Now, as pest control operators, you also have to be aware of what are some of the fleas that we see on the rats that we try to control, primarily roof rats and Norway rats. So this is the list of fleas. Very commonly, we'll see cat fleas on roof rats because cats and roof rats commonly occur in the same habitat in the suburban environment. We'll find stick-tight fleas on both roof rats and Norway rats. The oriental rat flea is the one that is most common in Florida, primarily because it is largely a flea of roof rats. It will also be found on Norway rats, but it is predominantly a roof rat flea. The northern rat flea is primarily a Norway rat flea. And also, at least in Florida, it is much less common than Xenopsilla chiapas, the oriental rat flea. And then again, we have the eastern squirrel flea, the European house mouse flea. And this one is actually one that we need to be aware of because this is the vector for rickettsial pox, Rickettsia acari. So as we go through the fleas, most of our pest fleas are in the family Pulicidae. The common name for that family is the common fleas. It includes the dog flea, Tenocephalides canis, the cat flea, Echidnophega gallinaceae, the stick-tight flea, Pulex irritans, the human flea, which is primarily found in the Midwest, Pulex simulans, which is primarily found in the Southeast, Xenopsilus chiapas, which occurs throughout the whole world, wherever you have roof rats found, has a very wide worldwide distribution. And then Silvolagus, the cottontail rabbit flea, Cydnocilla simplex. I'm not going to spend a lot of time on the rabbit flea because it's not really a concern to the pest control industry. All right, so this is the dog and cat flea. And you can tell a dog flea from a cat flea by looking at the shape of the head and also the teeth of the genial comb. That's the comb on the face. It looks like a mustache if you look at this flea underneath the slide. So the head is more rounded, more compacted, whereas cat fleas, their head is more stretched out. And then if you look at the teeth on the genial comb, the comb on their face, the first tooth is half the length of the second tooth in the dog flea. And it's the same length, both teeth are roughly the same length in the cat flea. So that's how you can tell those two species apart. Dog fleas, at least in the United States, are primarily associated with wild canids, especially coyotes, but to a lesser extent, red fox and gray fox. And then it gets on dogs that are sympatric or found in the same habitats as those wild canids, those foxes and coyotes. Cat flea, it is common on dogs and cats, opossums, raccoons, ferrets. I have seen them on squirrels, almost anything, any mammal that's in the suburban environment, very likely will have cat fleas associated with it. Now, again, I don't like the name human flea, but it is the name that's in the literature. We have two species of Pulex in the eastern United States. There's Pulex irritans, and then there is Pulex simulans, the southern human flea. For most of its known time, people didn't realize there were two fleas. They just thought all the fleas that looked like this were Pulex irritans. And it wasn't until the 1980s where they actually figured out that we actually have two species. And when I show you the range map, you'll see that there's a little bit of confusion on what the distribution is. So all the spots in yellow, those are known examples of Pulex simulans. The ones with the red spots, those are ones that are known to be Pulex irritans. And then all of those other spots are ones that were just identified as Pulex. And we don't know which one they were. So these are records from the literature and from museums. But generally, in the southeastern United States, it's going to be solely Pulex simulans or coastal perhaps. And then in the northern part of the eastern United States, then we're going to have mostly Pulex irritans, the true Pulex irritans. Why is Pulex important? It can sometimes be a vector, especially Pulex irritans, can be a vector for certain diseases, and it is easily confused with Xenopsilla. So that's the Oriental or the Brazilian rat flea or tropical rat flea. And the way you tell the two apart, because their overall appearance is very similar, is when you clear them and mount them on a microscope slide, there is a supportive rod of cuticle in the mesothorax. All right, so that's the middle part of the thorax. It's the second segment behind the head. And this supportive rod is only found in Xenopsilla, and it's found in all of the species of Xenopsilla that have been associated with plague, for example. And Pulex irritans and Pulex simulans do not have that supportive rod right here. So that's how we tell the two apart. And the reason it's important from a public health point of view is that Xenopsilus is a known vector for several diseases. And this is the oriental rat flea. Again, this is primarily going to be a flea associated with roof rats, although it will occasionally get on Norway rats. It is primarily a tropical flea, so it tends to be found in the warmer parts of the southeastern United States and throughout the world. So Xenopsilus chiapus, the oriental rat flea, it parasitizes all members of the genus Rattus, especially Rattus, the roof rat, although it will parasitize Rattus norvegicus and Rattus exulans, the Polynesian rat, as well. And because of its association with plague, especially as the dominant urban vector of plague, these are the fleas that distributed plague throughout Europe and resulted in millions of human deaths during the Dark Ages. Pulex irritans can also be a vector for plague. Now, the good news is that we don't have plague in the eastern United States. And nobody knows why. The CDC has been working on this problem for a long time. So plague got into the United States in 1900 in San Francisco, spread throughout the southwestern United States, primarily being maintained in sage voles and ground squirrels and prairie dogs, but it never made it east of the Mississippi River, and we don't know why. The vectors or the host of the fleas are there. We just don't know why plague has not shown itself in the eastern United States. Not that we're complaining. We still have 5 to 10 human fatalities a year from bubonic plague in the United States. Again, primarily in the Four Corners area of the southwestern US and going all the way into Southern California. We also have the flea-borne or murine typhus and cat fleas, as well as Xenopsilus chiapus and Pulex irritans, can be a vector for this disease. So this is caused by Rickettsia typhi, and it's vectored by fleas. And usually it's a rodent flea cycle and then spills over into human beings with Xenopsilus chiapus. But we also have a suburban cycle with cat fleas and opossum. We've seen this in Texas. Haven't seen it yet in Florida, but I wouldn't be surprised if it occurs. So those are some of the reasons why we have to be concerned about these particular fleas. So this shows you the distribution of the oriental rat flea. And sure enough, every place where we see large numbers of roof rats occurring, this is where we find this flea. So this is primarily a flea of the roof rat, although it will infest Norway rats to some extent. Right, this is the stick-tight flea. And usually you're going to see these on dogs and cats and birds. This is one of the few fleas that will feed on both mammals and birds. Very often we'll see them associated with poultry. They attach, unlike other fleas that bite, take a blood meal, and then move on. These guys will actually attach and will, over a long period of time, will take multiple blood meals. One of the ways to recognize this flea is if you look at the head where the nose would be, it has a very sharp angle. It almost looks like it's a square. That's one of the characteristics of echidnophaga. The other characteristic is that the thoracic segments are compressed. And so they're very narrow, and it gives this sort of scrunched up appearance to this flea. Now, stick-type fleas don't normally attack people. On birds, very often they're going to be associated with the face, around the eye, around the ear opening. If it's a bird like a chicken that has a comb and a wattle, they're going to be associated with those fleshy areas that don't have a lot of feathers. All right, now we're getting into another family. This is the Ceratophilidae. And the one that we're really going to be concerned about is Nosopsilus fasciatus. Yeah. Nosopsilus fasciatus is the northern rat flea. So this is the flea that is primarily on Norway rats. It will occasionally get on roof rats as well. And it tends to be more of a northern flea. So in New York City, with all of their Norway rats, this is going to be the most common rodent flea that they're going to have in New York City. And so this has a pronotal comb. So that's the one that's on the back edge of the pronotum, but it has no genial comb. And so that is pretty good, unique character. Remember that the Oriental rat flea doesn't have a genial comb here or a pronotal comb, similar to what we see with Pulex irritans and simulans. All right, so again, this one is more common in the northern part of the US and into southern Canada. And again, there's that pronotal comb and lack of a genial comb. All right, Leptopsilidae. All right, this family includes Scylla cygnus. This is the European house mouse flea. And this is going to be one of the very few fleas that you're going to find on house mice. It will also sometimes switch hosts, and you'll find it on roof rats and Norway rats as well, if you have them in the same structure as a population of house mice. So Leptopsilla cygnus, it has a pronotal comb. It's a fairly small flea, and it has a limited number of teeth on the genial comb. And instead of being horizontal, they're arranged vertically. And again, this can be usually on house mice, but it can also be on roof rats and Norway rats. And it is the vector for rickettsial pox, and the causative agent is Rickettsia acari. And this is what rickettsial pox looks like. So the good news about rickettsial pox is, compared to things like Rocky Mountain spotted fever and epidemic typhus and murine typhus, rickettsial pox tends to be self-limiting. So almost all people who are infected with rickettsial pox will recover even without treatment. With antibiotic treatment, they'll have a complete recovery fairly quickly. All right, so those are some of the important fleas that we need to be aware of. And one of the things that we want to talk about briefly is control. And since we're talking about control in residential areas, whether it is a single family home or multi-family structures, we're going to be talking about cat flea control. And so we're going to go through the parts of a cat flea control program. Detection and monitoring is going to be the first most important thing. And when you're doing the inside inspection and talking and doing your initial interview with client, the customer. You want to find out where their pets are spending most of their time. Some of the work that I did for my PhD, we actually found that there was a significant relationship between where the pets slept and where the majority of the flea larvae were located. So that's an important thing to find out from the customer. You could also go in and sample rooms by taking white cotton socks, pulling them up over your pant cuff and walking through the room. The reason you use the white socks is so that you can see the dark fleas running on the surface. Hopefully you won't take any of those fleas home to your family. For the outdoor inspection, you can also do the sort of the white sock test, walking around and see where you pick up the fleas, but find out again from the customer where their pets are spending most of their time, especially areas that are protected from rain, irrigation, and full sun. Full sun will dry out and kill the larvae. Heavy precipitation or irrigation will drown the larvae. And so that's usually not where the problem is, unless it's during the dry season. So find out where the pets are getting out of the sun and the rain, and that will tell you where you need to treat. So we normally are not going to have any contact with the pet. So checking the host, whether you're using something like a flea comb or you're checking for bites, which is usually done by the veterinarian, those are not things that you as a pest control operator should do. These are the responsibilities of the pet owner, and you really don't want any liability associated with doing anything with the pet. So you do need to make sure that the pet owner realizes that they have to treat the pet, either do it themselves or take the pet to the veterinarian, as well as treat the premises. Doing one without the other is usually not going to be effective. You have to do both things. Now, pet treatments where you actually are treating the animal should never be done by a pest control operator. It should be done by the owner or by a veterinarian. And the reason for that is if anything happens to that pet, even if it came down with an illness completely unassociated with anything that you've done, you're going to get blamed for it. It's not worth the liability risk. So you just need to make sure that the customers realize that is their responsibility. They are not, or you can't do that. Things like flea collars, you hardly see people use them anymore because the on-animal treatments have been so effective. The once-a-month treatments, the spot-ons, those have been very effective. So generally, flea collars are much less used. Now, if you're having problems with ticks, brown dog ticks, and you wanted to use a tick collar with Amitraz, that would make sense. But generally, flea collars are not going to be very useful. And you're not supposed to keep them on the animal all the time. I know this is what my mother used to do, where she would have a flea collar on the dog all the time. And most of the time, the active ingredient had already dissipated and it wasn't doing any good. But it is not healthy for the animal to be exposed for long periods of time to the chemical in the flea collars. It tends to cause irritation. So usually flea collars should not be used for longer than 6 days at a time. And they are not a prophylactic. So the homeowners should not use it as such. The other thing you have to be aware of is find out if they're using flea collars, find out what the active ingredient is, because you don't want to accidentally double dose the animal by using the same active ingredient that is in the flea collar. And I will go ahead and say this, and you'll hear me say it repeatedly. Ultrasonic flea collars do not kill or repel fleas. In the experiments that we did at the USDA in Gainesville, Florida, we actually found absolutely no effect, although there was a slight, and it could have been a sampling bias, but a slight increase in the number of eggs produced by fleas that were exposed to ultrasonic flea collars. But it was not significantly different than when the animal wasn't wearing the flea collar. And again, anything having to do with treating of the pet should be the responsibility of the homeowner or the veterinarian. As I said, the possibility of liability for a pest control operator is just too high. So there are different types of soaps, sprays, shampoos have been used to control fleas. When you comb out fleas, dump into a pail of soapy water, and you can actually remove all of the fleas from a cat. Probably the same for a dog, but it takes a long time, as Dr. Hinkle will be glad to tell you, because she did it repeatedly for some of her experiments. Once a month, flea pills or drops, these are available from the veterinarians, and it is their responsibility. Let them take the liability. But things like Program, Advantage, and Frontline, and then several of the newer products like have really decreased the problems that we're seeing with fleas in residential areas. It still tends to be a problem associated with feral cats and dogs. And these are some of the products that might be available to the homeowner. But remember, this is veterinary medicine. This is not pest control. And just some more examples. Now, what the pest control industry does is we can help with treating the premises. One of the tools that we use for treating is vacuuming. So a good vacuum with a beater bar brush can remove 15 to 20% of the larvae and 32 to 59% of eggs from carpeting. It almost will never remove any pupae because their silk cocoons are actually bound to the fibers of the carpet. Now, vacuuming will also help to remove some of the flea dirt. And flea dirt is that black material that you see on the pet or where the pet has been sitting, and that's actually flea feces. And that's the food for the larval fleas. So if you can reduce the amount of flea dirt that's present, the droppings of the adult fleas, it reduces the food supply for the larvae and might help to reduce the population. Now, if you can use a true steam cleaner, something like you might use for treating for bed bugs, this will kill all stages if you're thorough in your treatment. Flea traps, there are quite a few flea traps that are on the market, but realize that these are primarily monitoring tools that are used to detect the presence of fleas. And there are some that are better than others. Those that turn on and off seem to actually be more effective than ones that are continually producing a light source. But they're only going to be for monitoring and looking at the level of infestation you're dealing with. And again, the ultrasonic repellents are completely useless for flea control. Carpet treatments, this can be anything from a boric acid dust or Timbor, which is disodium octaborate tetrahydrate, better known as DOT, will kill larvae because it contaminates their food. They ingest it and it disrupts their midgut and they will die. So boric acid dust, if it is applied thoroughly, and unfortunately there's no good way to apply the needed amount which is about the size of, a quarter cup for an entire room, there's no good way to really evenly distribute that throughout the carpet. So that's one of the problems that we have with using boric acid or disodium octaborate tetrahydrate dusts, or any of the borate dusts, including borax. It will kill the larvae, but it is hard to distribute it in such a way that it's going to be 100% effective. There are some very useful insect growth regulators, however, for flea control. Things like methoprene and pyroproxyfen are extremely effective. We found methoprene is good down to about 4 parts per billion. Just a tiny amount will stop flea development in a structure. One of the good things about methoprene is that it translocates, it moves around, so you don't have to be quite as thorough. However, it is also not photostable. So after about a week, it's going to be mostly broken down, and you can't use it outdoors because it will simply break down in sunlight very quickly. Pyroproxyfen, sold as Nylar and several other trade names like Archer. This material is photostable. And in, again, some of the work that Dr. Hinkle did when she was working on her PhD with pyroproxyfen, we were finding that if you treat in like a crawl space where cats and dogs like hang out, we were finding up to 9 months of control with pyroproxyfen. So we have things like Precor, which is method, and then we also have some other products. Here's Nylar, which is pyroproxyfen. Thor, this is uses pyroproxyfen and tetramethrin and phenethrin. So it has 2 pyrethroids. I'm not sure why it has 2. And then it has the insect growth regulator pyroproxyfen. And then when you look at something like Alpine, which can be used indoors, it has dinatufluron, which is a neonicotinoid, pralethrin, which is a pyrethroid, and again, pyroproxyfen, which is the IGR. All right, lawn treatments. And this can be somewhat problematic because this is going to be one of those things where you're going to have to put a lot of pesticide, if you're using a traditional pesticide, over a large area of the landscape. So any place that the dog is going to run or cat is going to have access to. This earth has often been recommended for flea control. In the Southeast, it basically is useless because of our high relative humidity. Because diatomaceous earth is a desiccant. It causes the insect larvae, the flea larvae, to lose moisture and then die of desiccation. There are nematodes that you can use to kill larvae, and we've tested them. They work. Nematodes like Steinernema carpocapsae, you have to keep the area pretty wet with irrigation or pretty regular rainfall. And in those situations, the larvae are probably going to drown anyways. So you can't really tell if it's the nematodes that's killing the larvae or it's the excessive moisture. We have shown that the photostable insect growth regulators like pyroproxyfen can provide 9 months of 100% mortality in bioassays. So that's where we treated underneath in this case, it was a trailer. And then every month we took a sample of the soil, put flea larvae in it, and looked at their survival. And for 9 months, we were getting 100% control. For 12 months, we were still getting about 80% control. So diatomaceous earth doesn't work unless you're in California or the desert Southwest. Nematodes will kill larvae. I don't know if they will successfully kill the pupae in their cocoons. I don't remember anyone actually testing that, but pyroproxyfen sold under NAR or Surge or Pivot or Archer is very good for especially protected locations where the pets are going to spend a lot of time. It's going to be protected from rainfall, and you're going to get the most bang for your buck by treating those areas where the pets are staying under the shade or away from rain and irrigation. And if you're in a situation where you have a large rat population and there's concern about rat fleas, this is more of a problem out west than it is here, but there are products available that use both a rodenticide block with an insecticide. So these Adios and Kaput are two examples of those. And the idea is it not only kills the rats, but it will also kill the fleas that are feeding on the rats. Or for most pest control operators, this is a minor concern, but if you have a customer who is very concerned about fleas coming off of the rodents as they die, this might be something to consider. Okay, it has been just about 50 minutes. So before we get into ticks, we're going to go ahead and let's take a 5-minute break, and then we will start back at 4 o'clock. Okay.
Sounds good, Bill.
Dan, should we start up again?
Yeah, I think we're ready to go, Bill.
Okay.
Probably some people will trickle in, but you're ready to go.
Okay. So how bad was it? You guys get fed up with fleas? You didn't know there was that much to know about fleas, did you?
I think most people don't know the diversity of fleas. Who knew? How many flea species worldwide, Bill? Or Nancy, if you're online, what's the total?
Worldwide? We don't know.
1,000?
Oh, easily. And the thing is that because fleas are so host specific, it's entirely possible, because we're still discovering new rodents all the time. And even the rodents and critters that have been found in other parts of the world, whoever looks at the fleas.
Yeah. Nancy just chimed in. She says there's about 2,200 species worldwide at last count.
Okay. I was going to say about 3,000, but
yeah. Okay. Carry on.
All right. So now we're going to talk about ticks, everybody's favorite type of mite. All right. And we're going to start with the Lone Star tick, Amblyomma americanum. This is reported to be the most common tick in Georgia, except probably in suburban areas. The brown dog tick is undoubtedly the most common. The Lone Star tick is a significant public health threat because they transmit the pathogen that causes human monocytic anaplasmosis, and has been associated with southern tick-associated rash illness. And so far, they thought they had a causative agent for Starry, something called Rickettsia parkeri, but when they go back and they look at patients with Starry, very few of them actually have Rickettsia parkeri in their system. So we still don't know what the causative agent for southern tick-associated rash illness is, but it does seem to be associated with Lone Star ticks. These are sometimes called the seed ticks because they like to climb up on vegetation. And I remember that we used to run into these guys during the fall turkey or the spring turkey season. And you'd walk through them and you'd end up with 100 of them on your clothes because they were up on the top of the grass or the plants. The larvae and the nymphs primarily feed on small mammals and birds, but don't feed necessarily on rodents. So it's going to be things like rabbits. They probably will feed to some extent on other small mammals like shrews, but for some reason, they don't seem to be strongly associated with rodents. And then from April through August, we find them on large animals, deer and cattle, and also will bite humans, get on humans. And this is the tick that has been associated with the immune response that causes a food allergy to red meat proteins. That is really frightening to me. The fact that you could get bit by one of these ticks and suddenly never be able to enjoy a steak or a hamburger for the rest of your life is just horrifying. They have a very wide distribution throughout the eastern United States, as seen in the range map. The females are easy to recognize. They, on the back edge of their scutellum, they have this, it usually looks like a white spot, but sometimes it almost looks silvery, but they have this distinctive spot in the middle of the female's body at the back edge of the scutellum. The males, the scutellum covers the entire back. And so you don't have that lone star visible on the males. In Georgia, I understand they recently had a problem with bobcat fever, and this is actually probably the best name for it would be bobcat or cat malaria, because it is a hematoprotozoan parasite like malaria, and the vector is the lone star tick. And normally it is a bobcat lone star tick cycle, but if the ticks get on free-ranging cats, you can have anything from a subclinical carrier, which then can infect other ticks, which can infect other cats. You can have a cat become ill from this disease with a periodic fever, or it can be fatal. And sometimes I've heard that it can be fatal in a matter of hours. And this is what the parasite looks like at we call it the ring stage in malaria, but this is where they've gotten into the red blood cell and are multiplying. Gulf Coast tick, primarily along the coastal plain all the way from Virginia down through Florida and Texas. So basically the southern 2/3 of Georgia. This is a vector for the Rocky Mountain spotted fever. So it is the vector for Rickettsia rickettsii. And this is the one that they've also found Rickettsia parkeri in. And Rickettsia parkeri, they've identified the pathogen. They just haven't linked it to a disease yet. They thought it might be associated with Stari, but that doesn't necessarily seem to be the case. The larvae and nymph feed on small rodents and ground-dwelling birds. The adults feed on a large variety of mammals and human beings, cattle, deer, pigs, or feral pigs, dogs, and very often they occur in large numbers around the ears, especially behind the ears or in the ears. This is the female. That's her small scutellum. The males, the scutellum completely covers the back of the body. This is American dog tick, Dermacentor variabilis, the second most common tick in Georgia in non-urban areas or non-suburban areas. It is known to transmit the rickettsia that causes Rocky Mountain spotted fever and heartland virus in Georgia. Heartland virus was originally discovered in, I think it was Arkansas. And it is, once they started looking for it, they started finding it other places. It is still not widely identified. It can cause a encephalitis type of reaction, and so it can be very serious. The good thing about American dog tick is it takes at least 4 hours of attachment for them to transmit the disease. Larvae and nymphs almost exclusively feed on small rodents, particularly mice and cotton rats. And then the adults, again, are feeding on larger mammals, including everything from dogs and raccoons up to deer and feral pigs. Primarily, the adults are found March through September. This is the one, so this is the female, this is the male. And I always thought these sort of looked like castor beans. If you've ever seen castor beans, that's what these ticks reminded me of. Okay, black-legged tick or the deer tick. So this is the tick that transmits the spirochete that causes Lyme disease. It will also emit the bacteria that causes babebiosis and human anaplasmosis. So this can transmit multiple diseases. However, the one that we are most concerned about, especially in the northern part of their range. So this is essentially the range where people used to call these deer ticks. And down here in the southeast, we call them black-legged ticks. They are the same species. And this is the primary vector for Borrelia burgdorferi, the spirochete that causes Lyme disease. Larvae and nymphs feed on reptiles, birds, and some rodents. So in places like skink in the southeast, they're going to be feeding on skinks. They may feed on other types of lizards like fence lizards, but skinks are probably the most abundant lizards in the eastern United States. From September through May, larger animals, including cattle and humans and also deer, will be preyed on by black-legged ticks. And they're called that because they're dark brown color. This is the male. This is the female, again, with that shorter scutellum. And generally, the females are larger than the males. And that's true with most ticks. And especially when you see a huge gravid tick, it's always going to be the females because they get so big because they're producing eggs and their bodies are full of eggs until they lay them. Brown dog tick, this is probably the most abundant tick that pest control operators ever are asked to deal with. And it's actually a pretty easy tick to recognize. Rhipicephalus is identified by having a hexagonal basic capitulum. So the segment that the mouth part's attached to, okay, that is called the basic capitulum. It's the base of the mouth parts. And in virtually all of our other ticks, it is rectangular in shape. With brown dog tick, it's hexagonal in shape and it sticks out to the side. The one tick that you may confuse this with is Haemaphysalis, the rabbit tick, and then the introduced Asian longhorn tick. But for them, their basic capitulum is rectangular. And the flare comes out from this second segment of the palp. And that's how you separate Rhipicephalus from Haemaphysalis. There we go. And all of the ticks that we've been talking about so far are classified as 3 host ticks. So that means that the females feed on blood. They fall off, lay their eggs. Usually the female will then die. The eggs hatch. The larvae will get on a dog, feed on it, fall off, molt, get on another dog. Now, granted, this could be the same animal if it's in a household, but it will get on a dog, feed, fall off, molt, and get on a last dog. And this would be the adult, get on the last dog, the third dog, and take a big blood meal. And that's where you find those huge females that are completely engorged with eggs. So these are considered to be 3 host ticks. Most of our hard ticks are 3 host ticks. So this is the rabbit tick, Haemaphysalis, and this one is primarily found on rabbits, but it will sometimes get on some other things, usually accidentally. If they get on domestic rabbits, they are usually treated with ivermectin to kill the ticks because most of the compounds used to treat for ticks on cattle and horses and pigs and things would be too rough on rabbits and small mammals. And this has been shown to be a vector for Rocky Mountain spotted fever and is also the primary vector for tularenensis, which is tularemia or rabbit fever, right? These will almost, and I say almost, never bite people. And usually they are only found on rabbits. And this is the first segment of the palp is where you have this little shoulder sticking out. But you can see how if you're not real careful, don't look real close, you might think that you're looking at the basic capitulum. But you notice the basic capitulum here is rectangular. Now we have a new Haemaphysalis that has been introduced into the United States. This is the Asian longhorn tick, Haemaphysalis longicornis. And this is an invasive tick from Asia, and it is a parthenogenetic species. So at least in the populations that have been carried around the world, they are entirely female. There's no males needed for mating. They were first found in Georgia in 2021. And so far, we're not quite sure how bad of a problem they're going to be, but they are known in Asia. They are known as a vector for this bundavirus, but that virus is not in the United States. But there is concern that this could become a vector for the heartland virus. And that has been a concern. So it has shown up in northeastern Georgia, and these are some older site references. This map is showing potential range. So this is estimated based on habitat and climate data. So for those of you in Tennessee, Kentucky, Indiana, or Southern Indiana, Southern Illinois, Southern Ohio, West Virginia, and North Carolina, this is going to be a problem for you guys in the future. So it was first detected in Connecticut, and it's been spreading fairly rapidly. So again, Haemaphysalis, this is the basic capitulum. And then there's that first and palp segment, and it sticks out to the side like it's got big shoulders. All right. One last group of ticks that occasionally you might encounter are the argacid ticks or soft ticks. And Arga ornithobius are some of the ones that are associated mostly as problems with livestock, but occasionally you'll get them associated with people. They are found in animal shelters or in burrows, caves, and then occasionally you'll find them in poor quality human dwelling, people who are living in, let's say, mud and stick huts or moderately open wooden structures. You'll oftentimes have these ticks living in the cracks and crevices and coming out and biting people. Now, these are different in their biology than the hard ticks that we've been talking about. These guys are multiple feeders, and they will feed multiple times. And between feeding, they will leave the host, and they'll usually go and hide in the nest of the host or in the surrounding habitat. And then they come back out and take another blood meal. You could almost think of these guys like bed bugs in terms of their biology. Coming, taking a blood meal, going away. Coming and taking another blood meal, going away. So they may take multiple blood meals between each molt. And even after the female lays eggs, she will continue to take blood meals and produce multiple clutches of eggs. I've only seen this one time, at least in Florida, and it was associated with a home, and they had a pet pot-bellied pig. This was obviously a surrogate child because the pig had its own bed and its own room, and they noticed something was wrong when it stopped using its bed. It started sleeping in other parts of the house. So it can show up occasionally. And again, this is largely easily controlled with space sprays. And Nancy may want to jump in about it's a problem in livestock. I know it can be a problem in poultry. Or not. I don't think Nancy's listening to me. Okay. Tick control options are in treated clothing. We use this both for military uniforms, especially battle dress uniforms. So these are mass treated, and usually you can get 10 washings out of a single treatment. One of the questions I get quite often is, all right, we've got ticks on our dogs, but I don't want to spray a lot of chemical all over my yard where my dogs and my children play. So one of the suggestions I've made is use a tick drag, where you actually take a flag, or it could be a towel or whatever, treat it thoroughly with permethrin, let it dry, and then drag it over the ground. And that will pick up any larvae or nymph or adult that are present. They will grab a hold of it, become intoxicated, and die. And that way you're not applying lots of pesticide to the landscape. And that seems to be something that even chemophobes are willing to tolerate to control ticks. They're in treated nest material. And that's what this device is. It's basically a tube stuffed with cotton balls that have been treated with permethrin. And this was developed by Harvard, and it was for use in controlling Lyme disease in New England. And basically you just take these and spread them around the habitat, any place where cotton or deer mice or white-footed mice are likely to occur. They collect the cotton to use as nesting material, and it kills the larval and nymphal ticks. Indoors, pyrethroids, things like permethrin are still pretty effective. Products like Phantom, which is chlorphenapyr, or Zenprox, which is Fenprox, and PBO seem to be very effective on ticks indoors. Outdoors, permethrin is still widely used with multiple products. Demon WP, wettable powder, cypermethrin is a very hot pyrethroid. And oftentimes, and permethrin or deltamethrin or bifenthrin doesn't kill a pest, cypermethrin will. Imidacloprid is a neon, and then Suspend is deltamethrin. Advantage is S-fenvalerate. So these are you're mostly going to have the pyrethroids for outdoor use. For on-pet treatment, there is a tick collar that is available that uses amitraz. Amitraz is an interesting acaricide that only kills ticks and mites because of its mode of action. And so it is very safe for vertebrates, especially mammals and also birds. So that's another possible treatment method. So permethrin treated clothing, and I learned to really appreciate this during hunting season in Florida. I was up in a tree stand and I actually watched this tick that had gotten on me, and I'm sitting there watching it, and it's trying to get away from my clothing, and it's leaning back with its first 3 pairs of legs waving in the air, trying to get away, and finally it dropped off and died. Permethrin treated clothing is a very effective control measure for ticks, also for chiggers, and for mosquitoes, and to some extent biting midges as well. But this is a picture of a rag that's being used to collect the ticks in the environment and systematically intoxicate them with the permethrin without applying large amounts of permethrin to the environment. Okay, bed bugs. And this isn't a problem that we have in pest control, because I do the UF School of Structural Fumigation. As I tell people, bed bugs aren't a problem. Fumigate the house, it'll kill everything. but some people don't want to spend that kind of money. So then it's a matter of using spray insecticides and just being incredibly thorough. We have two primary species in the United States. In the subtropical and tropical zone, we have primarily the tropical bed bug. So Southern Asia, Central Africa, Malaysia, Indonesia, those areas are where we have tropical bed bugs, and then also all through the Caribbean. And we also have it fairly commonly found in Florida. And probably a lot of the Florida cases are from people coming back from vacation in the Caribbean. Throughout most of the rest of the temperate United States, temperate Europe, it is mostly going to be the common bed bug, Chimex lecticularis. So these are the two species. And to be perfectly honest, it really doesn't matter which species it is, whether it's tropical bed bug or the common bed bug. You're going to treat them both the same way. Neither of them is known to transmit disease. Okay, so one of the ways you can identify them is when you look at the pronotum, the front edge of the pronotum is sort of deeply dished out in common bed bugs and is more shallowly dished out in tropical bed bugs. Other than that, they're very similar to one another. Again, you're looking at the margin of the prothorax. So if it is steeply dished out like this, it's Chimex lecticularis. If it is very shallowly dipped out or curved, it's hemipterous. And considering how often people from around the United States go for vacation in the Caribbean, we may start seeing hemipterous showing up throughout the eastern United States, not just in Florida. Another species that we sometimes will encounter in homes is the bat bug. So this is Chimex adjunctus, the eastern bat bug, and then this is the common bed bug. And the easiest way to tell the two apart is look at the length of the hairs on the pronotum. If the hairs are not as long as the eye, not as long as the eye, then it is a bed bug. If the hairs on the edge of the pronotum are longer than the width of the eye, all right, then it is a bat bug. And generally, we don't have to be concerned about bat bugs. And this is showing it again. This is actually one of the Western bat bugs, Chimex estrella. And you can see how long those hairs are compared to the short hairs that we see on the common bed bug. Eastern bat bug, Chimex adjunctus, is primarily associated with big brown bats and evening bats. And generally, they are going to be feeding on the bats during the day when the bats are inactive. So bat bugs do not feed at night because that's the time when the bats are normally gone. So at night, they just crawl in their crevice and wait for the bats to return. And so that's one of the reasons why bat bugs are not as likely to be a pest as bed bugs. So this is normal activity pattern for bed bugs. So it starts at about 8 o'clock, really picks up after midnight, and then drops off before dawn. Okay, so this is the activity time for bed bugs when people sleep. Well, bat bugs are going to be feeding from roughly sunrise all the way through the day, and it's going to stop as we get close to dusk, because that's when the bats are going to start getting active and get ready to fly away. So the bugs will leave the bats and go crawl in a crevice. So that's one of the reasons why bat bugs are less likely to be a problem than bed bugs for people. Now, we do have some other species that show up. This is Orthochorus audis. It's sometimes called the Mexican chicken bug, but it actually probably got here from Brazil, I think. And so it has been reported from purple martin nests and also poultry houses in both Florida and Georgia. And it looks superficially a bed bug, but the pronotum is very different in shape, and it's almost going to be exclusively associated with birds. This is a bird feeding bug. But you may find it in people who keep backyard poultry. This is the North American swallow bug, and it feeds on cliff swallows in the western United States, barn swallows, and purple martins in the eastern United States. I don't know if it is associated with bank swallows, and I believe it's also associated with cave swallows in the western United States as well. This is known to be a vector of Buggy Creek virus and Venezuelan equine encephalitis virus, but only from bird to bird. And it's a pretty easy one to recognize because they are really furry. They're just covered with these hairs, dense hairs. And so this is another look at them. And you can see it looks distinctly different than a bed bug. So this is not something you would confuse with a bed bug. We also have one that's associated with chimney swifts. So this is actually a chimney swift nest in a chimney. This is what it looks like from the side. And these occur throughout all of the eastern United States, and then they migrate down through the Caribbean into Central and South America. But this bug sometimes is associated with chimney swifts. It's the chimney swift bug, Mexopsis nyctalis. And this, again, you might confuse this with a bed bug, but it's going to be associated with chimney swifts. So it's not likely to be a problem until you start seeing, or the chimney swifts migrate south. And then suddenly their food supply is gone and the chimney swifts start, or the chimney swift bugs start looking for other food sources. All right. We're almost to the end. The last 10 minutes, we're going to talk about venomous spiders and scorpions of the southeastern United States. So in the southeast, we have both the northern black widow and the southern black widow. These are native to the United States, to North America. Northern black widow extends up into Canada, and this is not the best map because northern black widow I have found as far south as Orlando in Florida. And we have both the southern black widow and northern black widow. Southern black widow, Latrodamectans, the females, adult females are completely black and they're shiny black. And they have a red complete hourglass on their abdomen. Males are much smaller than the females. They also have an hourglass. This is an immature female, so she still retains some of these juvenile markings that indicate a juvenile black widow. The egg sac is smooth and anywhere from a cream to a very light tan color, but it's smooth. The silk of widows is very tough, so that when you run your finger through a web, you'll actually hear the strands snap. It sounds like the snapping of monofilament fishing line. So they're very strong fibers in their web. So this is southern black widow, and you can see that red hourglass. Generally, these are cannibalistic, so you don't tend to find them in large numbers. At least with the northern black widow, if you find them on the eaves of a structure, you'll have one on one side of the building and one on the other side. And that's it. So these guys tend to be territorial, and they will kill any interlopers. So you tend not to have large populations of black widows, whether it's Mactans or Variolus. This is the northern black widow. Again, the adult females are shiny black, and they have a broken hourglass. It's still red, and it basically looks two red triangles pointing to each other. So that's how you separate the northern from the southern black widow. Northern black widows have a pretty wide distribution from Texas to Florida and then all the way up to Ontario. This lack of data here in West Virginia probably doesn't mean they're not found in West Virginia. It probably means they just haven't been reported. Now, both black or northern black and southern black widows are extremely venomous, and they are comparable in the severity of their venom. They have multiple toxins. So they have toxins that kill insects. They have toxins that kill crustaceans. So they're for things like sow bugs. This is one of the few spiders in North America that have a venom that affects vertebrates. And that venom will easily kill lizards as well as make human beings and other mammals very sick. The new one that we have to worry about is Latrodectus geometricus, the brown widow. In Africa, these are called brown button spiders. So they were originally brought from Africa to the Caribbean, and that was probably 350 years ago. They were brought to southeast Florida about 1900 or late 1800s. And for a long time, they just were restricted to a couple of counties in southeast Florida. During the time of Hurricane Andrew, when people were trying to get out of the way of Hurricane Andrew, everybody went north with their campers and their trailers and their boats, and they carried these spiders all over the eastern United States. So since Hurricane Andrew, they have spread all the way to Louisiana and Texas. They're at least halfway through Georgia. I found them in my farm in Ben Hill County in Georgia. So they are very widespread. They are continuing to spread. They can be recognized by this orange, kind of a hunter orange hourglass on the underside of the abdomen. The color variation is significant. Usually you will always see this banding pattern on the legs. The body, especially the abdomen, can be a marbled appearance. It can be a light color, almost an ashy white or gray, and all the way down to a satin black. So a lot of people confuse this brown widow with a black widow because of the texture. But if it's a satin black, it's a brown widow. If it is a shiny lacquered black, then it's probably one of the two black widows. So there's a wide variation of color, but they always have this orange hourglass on the underside of the abdomen. But what is easiest to identify them with is the egg sac. There is no other egg sac I know of in Florida or in the southeastern United States that looks like this. It's a spiky ball, usually tan in color. It's going to be about the size of the female's abdomen. And normally there's going to be anywhere from 60 to 120, 150 eggs inside. Okay. And a female will produce multiple egg sacs. And I've seen as many as 5 or 6 egg sacs in close proximity to a single female. Again, males are much smaller than the females. This is relative size. So this is a male compared to a female. both have the orange hourglass. Brown widows are slightly less dangerous. No, slightly less venomous than black widow. All of them have neurotoxic venom and will make you very sick. But brown widows in general seem to be less toxic. They are also less defensive. Usually when you disturb a brown widow, she just curls in a ball and drops to the ground. So she is less likely to come out and try to bite you if you mess with her eggs. So this is the current brown widow range. So it went from this area about 1990 when Hurricane Andrew hit to throughout the southeastern United States. Also, it's in Hawaii now. So it is widespread throughout all of Louisiana. It is spreading through Texas and through the Gulf Coast states. So this is one you need to be watching out for, and look for that egg sac. That is the most distinctive characteristic there is. Brown recluse, this is Loxosceles reclusa. And for those of you in Georgia and Alabama and Tennessee, this is one you need to be concerned about. Loxosceles it's a hunting spider rather than a web-making spider. So it will put out silk, almost like trip lines, so that when an insect comes by, it will touch the silk lines and then the spider can come out and grab it. So this is actually one that I photographed in Central Tennessee at the Mid-Tennessee Fire Academy. And there were a bunch of them, and they were all living underneath the garbage containers that were outside everybody's room. So brown recluse can be recognized partly by this violin shape on the cephalothorax, but if you have a dark individual, you're not going to be able to see that. But one of the characteristics they have is they have 6 eyes instead of the 8 we see in most spiders. And they occur in 3 couplets. So there's 2 eyes here, 2 eyes here, and 2 eyes here. The only other spider that comes close to having that arrangement is the brown spitting spider, but its body shape is completely different. But it also has 3 couplets of eyes. So this is the distribution of reclusa and its other relatives. Blanda is the blonde recluse spider in southwestern Texas. Devia is the one that's in southern Texas. Deserta is the one that is in the desert regions of California, Nevada, and Arizona. But for those folks in Georgia, especially North Georgia, virtually all of Alabama, Mississippi, Louisiana, Arkansas, Missouri, southern Illinois, southern Indiana, this is going to be the habitat for Loxosceles reclusa. And we always get people telling us that they've been bitten by a brown recluse in Florida, and we tell them, not unless you were someplace else. Now, another species that has been showing up, and this has become a worldwide pest. So this is one of those tramp pests that gets carried from place to place. This is the Mediterranean recluse, or sometimes it's called the Mediterranean brown spider, Aphelis rufescens. And this one seems to be associated with larger buildings. I know that it's been seen in large municipal buildings or government buildings throughout Washington, D.C., throughout Atlanta. I know that we found it in Miami, again, but in large buildings and usually in the basement. There seems to be a hypothesis that this was actually a cave-dwelling Loxosceles. And so it is going to look for habitats that are reminiscent of a cave in the Mediterranean region, both North Africa and then Greece, and Italy. It does have 6 eyes. It does have that violin shape. It tends to be a lighter color than reclusa. But there are some characteristics you can use to identify it. But for most part, it doesn't matter. If you identify it as a lock, you're going to go ahead and you're going to want to treat to control it. So that can either be something like a space spray or a general crack and crevice treatment in the areas where it's found. We've had one case of Loxosceles lata, the Brazilian recluse spider in Florida, and that population was controlled by fumigating the house. So this is the kind of the greenish area, or the yellow area is where these have been found or are expected to be found. And if this map, the small map, actually shows global records. So we have a lot of them throughout the United States. We have them out in Hawaii. We have them throughout the Mediterranean region and also Southeast Asia. And we actually expect that China is going to be have a significant problem with this. We have been seeing them along the coast of Australia. We expect to start seeing them along the eastern coast of South America. But look at the distribution in the United States. So any large city in the eastern United States is a potential source for this Mediterranean brown spider, Loxosceles rufescens. All right, real quick. Now, in the Southeast, we only have 2 species of scorpions. There's the southern devil scorpion, the Hovus carolinensis or carolinianus. And so it is found central Georgia, central Alabama, Tennessee, North Carolina, into Kentucky. It is about 1.5 inches long and it's brown. None of the spiders in the eastern United States are dangerous to people. That doesn't mean it doesn't hurt when they sting you. They're just not dangerous to us. The second one is Hensis centroides. This is Centroides hensi, sometimes called the little striped bark scorpion. Again, about 1.5 inches long, and their distribution is throughout the southeastern United States from about Anna to Mobile, and then all of Florida. And then there's also a population up here around Charleston. And if you look at the distribution, you can see how Hensis centroides does not co-occur with the Carolina devil scorpion. And again, both of these are not dangerous to people, but they can be painful. The easiest way to inspect for scorpions is to use an ultraviolet light because they fluoresce. And in places where Centroides hensi occurs, I've gone out with a black light, and every single pine tree had a scorpion on its bark. Only one per tree, but every single live pine tree had. And that's it. Are there any questions? And I'll leave this up in case you want to contact me for more information.
That was really good, Bill. We'll have to have you back next year. Talk maybe either elaborate on some of this or have you back for vertebrates. That would...
I have a whole talk all ready to go on the venomous Hymenoptera of Georgia.
Oh, that'd be nice. Do you have any questions, folks? I did have one question, Bill, about it. This is for you and Nancy, if you're still there. I get questions sometimes from people. You were talking about distribution of flea larvae, of course, being associated with host distribution. And I get calls from people who swear they have fleas in their yard. And I can only think they're talking about a distribution of adult fleas in the yard. And I can't get my mind around, or I just don't, you know what I'm talking about. I just don't,
right.
Fleas, when a normal distribution in the yard is just not, I don't know if they're springtails or what's going on.
These are true fleas, almost always cat fleas. And it's because you have the animals, either dogs or cats, that are spending a lot of time in the landscape. And in Florida, it's usually going to be in the springtime during our dry season, because as soon as we start getting our heavy summer rains, most flea infestations in the landscape are going to end because the larvae are going to drown, or their food's going to get washed away. And it's usually associated with where the animals spend their time. So a lot of times it's going to be under the shade of a tree, and the flea eggs and the flea feces are dropping off into the landscape, into the sand, and the larvae are going to have plenty of flea feces to eat. They'll develop and pupate, and then when you walk through the yard, your activity is going to disturb them. They're going to emerge from their cocoon and jump on you.
And Nancy has another good point here too. A lot of people have steps going up to their front porch or whatever. And of course, that's where the dog hangs out in the summertime. As you're walking up and down your steps, if you mingle around your steps, you're going to get fleas on your, and then you walk out in the yard and you think you have fleas all over you. But that's a good point. Had one question here, Bill, about spider mortality in terms of visual treatments. So the question is it true the only way spiders die from a product is from direct spray, being that they don't clean themselves like cockroaches? So it's an exposure issue. Can you comment on that?
It depends on the species. When I was doing some spider work for bear, and what we found was that we were getting pretty good control by treating surfaces. And we were using some dual active ingredient products. So usually it was a neonic and a pyrethroid mixed together. And we were getting 4 or 5 months of control. And basically after treating, we didn't have any reinvasion in our sites. For some spiders, they will try to recycle their silk. And that would be how they would come in contact with the poison. If it is a spider that doesn't spend a lot of time in its web or is going to be hidden in a crevice, and usually if you do a good thorough treatment of the environment, you will have good control of spiders. Some people claim that dusts are more effective, but if you use a microencapsulated product or a wettable powder, that will work just as well.
How about, I had a question of Bill about a bat bug. So when we do see a bat bug, we typically get an adult, which are a lot more mobile than immatures, and it's typically in a really odd area, be in the middle of the floor somewhere or a windowsill. To be in an area where there's no evidence of bed bugs, and oftentimes bats have been excluded from the structure. How long will a bat bug live, and will they readily feed on humans? How long do they live after an exclusion, a bat exclusion?
I'm going to say it's going to be probably about the same amount of time as a bed bug. So was it normally 6 to 9 months if they can't find a blood meal? And I guess if they get desperate enough, they will bite people. But remember too, like I said, their activity pattern is wrong for biting people. They're actively looking for hosts during the daytime, which is when the bats are normally going to be asleep, and usually where we're not going to be around. So
I was going to say they're probably attracted to CO2.
Yes, they probably are. They're probably attracted to the same things that bed bugs are. And remember that the bed bugs, all 3 species of bed bugs originally were parasites of bats. And so they switched over from bats to people maybe as far as 10,000 years ago.
Yeah.
But actually bed bugs will still feed on bats. They will also feed on birds. The very first samples of bed bugs I ever received were in the late 1980s. And it came from somebody who had cockatiels, and the bed bugs were living in the perch, in the hollow cardboard perches of the cockatiel cage. And they couldn't figure out how they couldn't get them under control. And that was the first time I'd ever seen bed bugs. I'd collected bat bugs lots of times during my bat research, but that was the first time I'd ever seen bed bugs. And then, of course, since the 1980s, their populations have exploded, not just in the United States, but worldwide.